Today I'm going to share on the topic Plasmodesmata, its structure and transport, which comes under plant biochemistry, and myself is Priya N from the Department of Biochemistry, St.Joseph College of Arts and Science, Cuddalore. So this plasmodesmata, which is a singular plasmodesma, which are small channels that directly connect the cytoplasm of neighboring plant cells to each other. That is, it acts as a bridge between the two cells, establishing living bridges between two cells. As I have telling you that it is act as a communicating channel between the two cells. So that bridge is called as plasmodesmata. And similar to the gap junctions, which is found in animal cells, this plasmodesmata will penetrate both the primary and secondary cell walls, which allows molecules to pass directly from one cell to another and are important in cellular communication. As I have mentioned you earlier, it mainly involves in cellular communications. What it happens means that communication means only the molecules transfer from one cell to another for nutrients, for the development of the growth. For that purpose, this plasmodesmata plays a vital role. And this is the intracellular junction of plasmodesmata. The parts of the plasmodesmata will be smooth endoplasmic reticulum, middle lamella, primary cell wall, desmotubule, cell wall of adjacent cells, cellulose microfibrils, pectin, and then cross-linking glycan and plasma membrane. So these are the parts which is found in the plasmodesmata. The plasmodesmata is structured in an entirely different configurations than the animal cell gap junction because of the thick cell wall. So, due to the presence of plasmodesmata, plant cells can be considered to form a syncytium or multinucleate mass with cytoplasmic continuity. So it was somewhat cylindrical in shape, and plasmodesmata aligned with the plasma membrane, as I have mentioned you in the structure. So all connected cells are united through essentially one continuous cell membrane. So the majority of the plasmodesmata also contain a narrow tube-like structure called desmotubule, which is derived from the smooth endoplasmic reticulum of the connected cells. In this plasmotubule, that desmotubule does not completely fill the plasmodesma, and consequently a ring of shared cytoplasm is located between it, and the inner surface of the membrane is lined with a channel. And this plasmodesmata typically form during cell divisions, when parts of the endoplasmic reticulum of the parent cell get trapped in the new cell wall that is produced to create a daughter cell. So transport mechanism of plasmodesmata. So transport is either by two way. One will be by apoplastic pathway and other will be by symplastic pathway. Apoplastic pathway, it is through cell wall, and symplastic pathway, it is through cytoplasm. Let's see. Symplastic transport is mediated by the plant specific structures called plasmodesmata, which are plasma membrane lined pores that transverse the cell walls of adjacent cells, thus connecting their cytoplasms. Then traffic via plasmodesmata is highly regulated. The accumulation of plasmodesmata is highly regulated, and callose turnover is known to be one mechanism for this traffic flow. Then thousands of plasmodesmata may be formed that connect the daughter cells to one another. And it is widely thought that by constricting and dilating the opening at the ends of plasmodesmata, plant cells regulate the passage of small molecules such as sugars, that is for their nutrients, salts, amino acids through this mechanism. So it can transfer nutrients through this process, through this desmata, through this membrane. That is molecules which is act as a nutrients for the growth of the cell. Then it is known that in some cases the size restrictions on molecule passage between cells can be overcome by binding to parts of the plasmodesmata. Special proteins and some viruses are able to increase the diameter of the channels, which is enough for unusually large molecules to pass through. Then structure. The plasmodesmata plasma membrane, its structure may be between 10 to the power 3 and 10 to the power 5 plasmodesmata, and approximately 50 to 60 nm in diameter at the midpoint, and it has three layers, that is plasma membrane, cytoplasmic sleeve and desmotubule. They can traverse cell walls that are up to 90 nanometer in thickness. Then the plasma membrane portion of the plasmodesmata is a continuous extension of the cell membrane or plasma lemma and has a similar phospholipid bilayer structure. Then the cytoplasmic sleeve is a fluid-filled space enclosed by the plasma lemma and is a continuous extension of the cytosol. Then tracking of molecules and ions through plasmodesmata through the space, as I have told you earlier. Then the molecules will be sugars, amino acids, ions. That will be done by diffusion method. Larger molecules including proteins, that is green fluorescent protein and RNA, can also pass through the cytoplasmic sleeve by diffusion method. So one mechanism of regulation of permeability of the plasmodesmata is the accumulation of polysaccharide callose, that is around the neck region, to form a collar, thereby reducing the diameter of the pore size which is available for transport of substances. And through this dilation, active gating or structural remodeling, the permeability of the plasmodesmata is increased, and this increase in plasmodesmata permeability allows for larger molecules or macromolecules such as signaling molecules, transcription factors and RNA proteins complexes to be transported to various cellular compartments. And desmotubule. It is a tube of appress, that is flattened endoplasmic reticulum, that runs between two adjacent cells. Then around this desmotubule, plasma membrane areas of an electron dense material have been seen, and it is often joined together by spoke-like structures that seem to split the plasmodesma into smaller channels. And these structures may be composed of myosin and actin, which are part of cell cytoskeleton. If this is the case, these proteins could be used in the selective transport of large molecules between the two cells. And plasmodesmata have been shown to transport proteins. So these are all the things which I have been mentioned you earlier. And it is used by the cells in phloem and symplastic transporters used to regulate the sieve tube cells by the companion cells. And the size of the molecules can pass through the plasmodesmata as determined by the size exclusion limit. And this limit is highly variable and is subject to active modification. For example, it is 30 is able to increase the size of the exclusion limit from 700 daltons to 9400 daltons, that way aiding its movement through a plant. Likewise, it is suggested that such a transport is mediated by interactions with proteins localized on the desmotubules, by chaperones, partially unfloated proteins, allowing them to fit through the narrow passage. And similar mechanism may be involved in transporting viral nucleic acid to plasmodesmata. So this is the callus formation, which is formed at the neck of the phloem. The plasmodesmata callus, it has a beta-1,3-glucan polymer which is known as callose, and it is found in cell plates during the process of cytokinesis for the completion of the level of Gall's. And in order to regulate what is transported in the plasmodesmata, callose may be present. So this callose provides a mechanism in which plasmodesmata permeability is regulated in order to control what is transported between the two different tissues. And this plasmodesmata undergoes several specialized conformational changes. And they also have a pattern of environmental, physiological and developmental cues that show relation to plasmodesmata function. And the enzyme activities of beta-1,3-glucan synthase and hydrolases are involved in changes in plasmodesmata cellulose level. Some extracellular signals change transcription of activities of the synthase and hydrolase. Then functions of plasmodesmata. Signaling and transport mechanism of plasmodesmata, as I told you earlier, for macromolecules it is by active transport, and in micromolecules it is by passive transport. And other functions of plasmodesmata will be transport gibberellic acid in plasma, sensitive to various levels of calcium, pH and light, control of turgor pressure, and initiation of wound response in plants. Thank you.